everyone, today we are going to study about gram positive and gram negative bacteria, the difference between the gram positive and gram negative bacteria. How can we identify which one is the gram positive and which one is gram negative bacteria? Okay, so we can identify the gram positive and gram negative bacteria on the basis of their response to a staining method called gram stain. Okay, so this is a technique which was developed by a Danish bacteriologist named uh, Hans Christian Gram. Okay, so the classification of these bacteria is based on their structural and chemical composition of their cell wall, which affects the staining properties. Okay, so as I told you in my previous video lecture, that there is a peptidoglycan layer in their cell wall. Okay, so this peptidoglycan layer retains the stain. I will explain this technique to you. So let's study the technique. Okay. So what happened in the first step? We prepare a bacterial smear. Okay. We take a bacterial sample and spread it on the slide gently. Okay. Then we air dry it. After that, we stain it with a with crystal violet. Okay, which is we called primary state. What does the crystal violet do? Actually, it makes the cell bluish purple in color. Okay, it makes bluish purple in color. Okay, so what we do after it? We rinse the slide gently, and then we treat it with another stain which is iodine, and we called it a mordant. Why we use iodine? Actually, this step is required to enhance the retention of stain. Okay, so we stain it with iodine and wait for 1 minute. Okay, after this, the another step is next step is decolorization. What we do in to decolorize the agent? Actually, we treat it with ethanol or acetone. Okay, so to decolorize the stain. So this is a very important step, because after that some of the bacterial cell get stained and some do not get stained. Okay, so if a bacteria retain the stain with bluish purple color, then it is gram positive. Okay, but if it get de-stained, okay, means no bluish purple color, then it is a gram negative bacteria. Okay, so after that we give a counter stain which is saffronin. Why we give a counter stain? Because when some of the bacteria get de-stained, means they get decolorized, so they take up this counter stain saffronin, which makes the bacterial cell pink in color. Okay, so when we rinse and bloat with a paper and rinse it gently and bloat it, after air dry it and observe it under microscope, we see some of the bacterial cell in bluish purple color and some of the cells in bacterial cells in pink color. So we can now make conclusion that the bacteria which looks bluish purple in color are gram positive, and the bacterial cells which looks pink in color are gram negative bacteria. So this is all about the staining technique. Okay, so why actually uh, the gram-positive bacteria retain crystal violet because they have a thick peptidoglycan layer. Okay, they have a thick peptidoglycan layer. Also, they have teichoic acid that contributes to the rigidity and overall cell wall structure. While in gram-negative bacteria, they have a thin peptidoglycan layer in comparison to the gram-positive bacteria. Okay, so instead of crystal violet, they take up the counter stain, which is safranin. Okay, so now look at the differences. First one is based on the cell wall structure. So gram-positive bacteria possess a thick layer of peptidoglycan, while gram-negative bacteria consist of a thinner peptidoglycan layer. Okay. Next is staining color. Obviously, so gram-positive bacteria, because of a thick layer of peptidoglycan, retain the crystal violet stain during the gram staining technique and appear purple or blue in color, while the gram-negative bacteria take up the counter stain, which is safranin, instead of crystal violet and result in pink or red color. Okay, the next step is outer membrane. So mostly the gram positive bacteria does not possess an outer membrane, which I told you in my previous lectures, which is the outer membrane is the glycocalyx. Okay, so gram negative bacteria has an outer membrane. They have peptidoglycan layer also, but they have outer membrane also. Okay, so which gives this characteristic to gram negative bacteria of staining. Next is antibiotic susceptibility. So gram-positive bacteria are more susceptible to antibiotic, but gram-negative bacteria offers resistance to antibiotics because of the presence of outer membrane. Okay, toxins. On the basis of toxins, gram-positive bacteria generally produce few endotoxins, but gram-negative bacteria produces endotoxins due to lipopolysaccharide. Lipopolysaccharide is present in the outer membrane because gram-negative bacteria have outer membrane, okay, which contain lipopolysaccharide. Okay, next examples are Staphylococcus, Streptococcus, Bacillus are the examples of gram-positive bacteria. E. coli, Salmonella, Pseudomonas are examples of gram-negative bacteria. So this is all about the gram-positive bacteria and gram-negative bacteria. Thanks for watching.